Hi, everyone. Welcome to our PacVec seminar series. Celia is off today, so I'm filling in for her as the host. And today we've got two great talks coming up from two of our training grant recipients in the past. So we'll get started right away. If you would, just hold your questions and feel free to type them in the chat if you like during the talk, but just don't forget them. And so at the end of each talk, we'll have the opportunity for you to ask questions by either unmuting yourself, or we can read them from the chat if you prefer that. So first up, we have Molly Bechtel, and Molly is working as a desert tortoise biologist with the USGS, and she graduated from another academic institution in our region, the University of Nevada, Reno, with a bachelor's and master's in animal science. Currently, Molly is a PhD student in Dr. Jeffrey Foster's lab at Northern Arizona University, and she's studying tick-borne disease, as you'll hear about, in desert tortoises and how the ticks and disease interact with the Mojave Desert burrow ecosystem, and potentially how they may matter also for public health. And so her current work has led her to become increasingly interested in how vector-borne diseases and wildlife populations relate to public health and the concept of One Health, which is animals, humans, and ecosystem health all as one concept. And she's planning to continue research in vector-borne disease interactions in wildlife and potential relationships with human populations. And in her free time, she founded and volunteers with the Northern Arizona Association for Women in STEM, which advocates for equal representation of women in the sciences. So with that, Molly, take it away when you're ready, and you can share your screen, I believe.
Okay, thank you for that introduction. Yeah, let's see here. Let me share my screen. Says it's still disabled.
Let's hope I can do that. Now it should work, I hope.
Yep, okay. Okay, share. Okay, just a minute while I get my presentation up. Okay, can everybody see that?
Looks perfect.
Okay, good. All right, yeah, thank you for that introduction. So I'm going to talk a little bit more about my research today on ticks and tick-borne pathogens of the Mojave Desert. But first, I want to give some background on the Mojave Desert tortoise. So some of you may know, some of you may not, that the Mojave Desert tortoise is actually listed as threatened by the U.S. Fish and Wildlife Services. It has been since 1990. And this is for a myriad of reasons, including disease, but also habitat fragmentation and climate change and things like that. But regardless, because the populations are declining, they are monitored per government regulation. And what I did when I worked at the USGS as a desert tortoise biologist is I did participate in some of these health assessments. And oftentimes we would notice ticks on these health assessments. But aside from just noticing them, just writing down how many we saw, and then picking them off the tortoise, that's all we really did with them. So let's talk more about these ticks. So the ticks that we find on tortoises are a soft tick. They're species of in the genus Ornithodorus. And there are two species of Ornithodorus that occur on these tortoises. So Ornithodorus parkeri and Ornithodorus turricata. There's not a whole lot known about them and their relationship with the desert tortoise. In the Mojave Desert especially, they're kind of overlooked. And in the literature, they're often just referred to as tortoise ticks because they like to spend so much time in tortoise burrows and on tortoises. So these are soft ticks. So their behaviors, their morphology is a bit different from hard ticks that most people are familiar with. These guys are nidicolous, meaning that they like to be in dark areas like nests. So actually desert tortoise burrows make perfect habitat for these guys. They're also generalists, which means they don't really care what kind of animal you are. So long as you have blood, they will happily take a blood meal from you. They're not specific to one species. And as I mentioned, they commonly parasitize desert tortoises. These guys are also difficult to differentiate between the two species. So between Ornithodorus parkeri and Ornithodorus turricata, the way to tell the difference between them morphologically under a microscope is basically by the number of bumps on their backs here called mammillae. And that can be subjective sometimes when you're looking under a microscope and counting. And oftentimes it's impossible to speciate them if you have nymphs, for instance, very small ticks or larvae, then you can just about forget about it. So these guys are also vectors of tick-borne relapsing fever group Borrelia, which causes tick-borne relapsing fever in people. And so there's 2 species of Borrelia that are going to occur in these vectors, so respective to the vector. So Borrelia parkeri is going to be carried by Ornithodorus parkeri, and Borrelia turricata is going to be carried by Ornithodorus turricata. And just to talk a little bit more about these Borrelia species, tick-borne relapsing fever group Borrelia occurs worldwide. They're carried by soft ticks with one exception, but I'm not going to talk about that today. So what I'm going to focus on here, this is the group that I'm studying, are these strains of Borrelia, Borrelia parkeri and Borrelia turricata, which do occur in the Mojave Desert and are carried by their respective tick vectors. Borrelia hermsii also occurs in the West. In fact, that's where most of the data comes from on tick-borne relapsing fever, is from Borrelia hermsii and its respective vector, Ornithodorus hermsii. But I'm going to focus on this group right here, which occur in the Mojave Desert and their vector, commonly parasitized desert tortoises. So with tick-borne relapsing fever in people, so note that desert tortoises are just susceptible to being, well, actually, we don't know. Part of my research, as far as we know, they do not get infected and come down with clinical signs for tick-borne relapsing fever, but people do. So what happens is when somebody gets bit by a tick that's carrying this Borrelia spirochete, this guy right here, this is a Borrelia turricata spirochete, about a week after being bit by that tick, they come down with really high fever, flu-like symptoms, aches. You just don't feel very good. And that lasts for about 3 days, and then it goes away for a week. And then it comes back, and then it goes away for about another week. And these cycles can actually continue until eventually they can just dissipate on their own, but they're very easily treatable with doxycycline. That being said, though, oftentimes if it's left untreated, it can lead to more serious complications and lead to hospitalization. So it's good that folks are aware that this is an issue in the desert, in the Mojave Desert, and in areas like Las Vegas, because it's just better, it's easy to get taken care of. And these relapses, so the reason that there is only a few days of fever followed by a week of nothing is because the spirochete has the ability to vary its antigens and in that way evade our immune system. It's kind of a clever little guy in that respect. But as far as Borrelia estimates are known in tortoise ticks in the Mojave Desert, but there's really nothing known about that. What we do know is from earlier data. So in the Aedes, Green did some work on desert tortoise and their associated ticks and found that about 10% of wild desert tortoises were parasitized by ticks in the samples that he collected, and about 43% of active tortoise burrows were infested with Ornithodorus parkeri. Again, all of his work was done with microscopy. So even the fact that he says, well, it was mostly Ornithodorus parkeri is debatable in some of the literature because it's so difficult to be able to differentiate the 2 species that we know occur in the desert. At any rate, this brings me to my research question. So I'm interested in learning more about this relationship between these ticks and the tortoise, and also more about pathogen prevalence and Borrelia that is occurring here in the Mojave Desert. So first, I collected over 1,000 ticks. I went out in the desert myself, but got a lot of these with the help from other tortoise biologists who were working in tortoise burrows or with actual tortoises, and collected them and sent them to me. And I looked at them under a microscope to determine their species. So I did find mostly Ornithodorus parkeri, which seems to be consistent with the literature. Everybody seems to be finding Ornithodorus parkeri in the Mojave Desert in Nevada. And about 206 of these were Ornithodorus turricata. So I mentioned earlier that microscopy can be really difficult when trying to speciate these guys. And in fact, over 500, I couldn't ID to species because they were too small, or the number of mammalae didn't quite add up. Again, there is some subjectivity here. Also, I did find one Otobius species, which is a soft tick, but not an Ornithodorus, and is mostly associated with rabbits. So because of the ambiguity that we have with microscopy and IDing these ticks, I have started sequencing them, which has been really interesting, just to follow up with that microscopy work. And so far, I found 6 ticks that sequenced to Ornithodorus terracota and 236 that were sequenced to Ornithodorus parkeri. And so far in these samples, I haven't found any pattern as to where Ornithodorus terracota has been found or versus Ornithodorus parkeri has been found. And I still have quite a few more ticks to run through. So to learn more about this relationship also, I took those health assessments that I mentioned earlier that the government requires when we monitor desert tortoise populations. And I looked at tick presence versus variables such as location, sex, signs of disease. And what I found was that adults are more likely to have ticks present on them than juveniles. Tortoises with clinical signs of disease are more likely to have ticks present on them than tortoises without any clinical signs of disease. Tortoises that have no forage evidence present are more likely to have ticks observed on them than tortoises without any forage evidence present. So just quickly, how you determine forage evidence is, well, sometimes as you're out in the field and you can just watch the tortoise eat, but they'll also have this fresh green staining on their beaks. So that's how you determine if there's forage evidence or not. But again, if a tortoise didn't have any of that staining on their beak, they were more likely to have ticks observed on them than those that did. And then finally, I looked at location. So I compared wild sites. So these are tortoises that are out in the desert at critical tortoise habitat locations where these populations are monitored. And I compared those sites to captive sites. Captive sites are, again, where tortoises are going to be living in a little paddock and only have one burrow. So I found that tortoises living in captive sites are much more likely to have ticks observed on them than those that are in the wild. Okay, and I should mention that none of this analysis is really indicative of disease either. So this is just based on those variables that are used in those health assessments. So what does this tell us about the biology of the tick and the tortoise? So you could say these Ornithodorus ticks are inherently lazy, but actually they're really well adapted to harsh environments like the desert. So they know better than to leave the burrow, this nice comfortable burrow, and go and quest, for example, for a meal. So they'll happily stay in a burrow or a dark nest for as long as it takes for a meal to come to them. Again, a really great adaptation for living in the Mojave Desert. So then it makes sense that tortoises that have a higher site fidelity are tied to one burrow more than others are going to have a higher likelihood of getting ticks. And that's going to include those adults. Tortoises who are, as opposed to juveniles. Juveniles can't really dig burrows yet. They like to stay under dark veg and maybe even just make a little bit of a pallet. So they're not going to be as likely as to be exposed. Same thing with captive tortoises. Captive tortoises only have one burrow, so they can't really, and they're in a small paddock, so they can't really go and explore the desert or go to another burrow. Even tortoises with more clinical signs of disease, they just may not be feeling so hot. So they're going to hang out more in their burrow than they are going to be, tromping around the desert. And speculatively, tortoises with forage evidence, well, they have been out foraging, out in the sunlight. And these ticks don't like that. Like I said, they like dark areas. So those ticks will have an opportunity to jump ship as soon as that harsh UV light hits them, whether it's at the mouth of the burrow or it's under some dark vegetation that the tortoise has been hanging out around. So it also kind of makes sense that people who are around tortoises and in desert tortoise habitat are exposed to these guys. In fact, with the help of PacVec, I recently just published this paper that documents the first 2 cases of tick-borne relapsing fever that occurred in association to these ticks. So 2 tortoise biologists did come down with tick-borne relapsing fever. In both cases, they were digging up tortoise burrows. So case 1 occurred out at a wild site, and case 2 occurred out at a captive site in which both of these biologists were digging up burrows. Interestingly, this second case, she was aware of tick-borne relapsing fever having occurred in the Mojave Desert, and was trying to explain this to her doctor. And it took forever to get a test to actually test for tick-borne relapsing fever group Borrelia, because they kept saying it's not a risk in the Mojave Desert. It's not a risk here in Vegas, but it is. So knowing that tick-borne relapsing fever and Borrelia is there, it is there in the desert, I took my 1,000 samples of ticks and I tested them for tick-borne relapsing group Borrelia. The prevalence was low, so about 22 tested positive for tick-borne relapsing fever, 5 of which I was able to sequence to Borrelia turricata. Okay, so still a risk, but the prevalence is really low, which is really interesting to me, considering that previous studies have looked at reptiles and this really interesting relationship between ticks. So generally, if a tick is carrying this Borrelia, this bacterial spirochete, it comes up to a person and bites it. Well, bites that person, they're going to contract tick-borne relapsing fever. But more recent studies, including one that was just published in Science in February, I believe, speculates that, well, it's reptiles and lizards that are keeping this prevalence so low of keeping Borrelia prevalent so low in the desert. And I'll explain why. So there was a study done in the 1990s by Bob Lane at Berkeley, in which he looked at these guys. This is a Western fence lizard here, and you can see there's some ticks hanging out on him. So he wanted to know more about the relationship between these ticks. And these ticks, they're hard ticks, Ixodes pacificus, and they carry Lyme disease, which is also a species in the genus Borrelia. And what he found was that these lizards have what's called a borreliocidal factor in their blood, which actually kills Borrelia. So what happens is this tick, who's carrying this Borrelia spirochete, comes up to take a blood meal from this lizard. Well, that borreliocidal component in the blood cleans the tick of that infection and is no longer capable of causing disease in people. So that's really cool. So then another study done in 2010 by Dan Salkeld looked at the abundance of lizards versus nymphal infection of Borrelia. And he found that where there are more lizards, there's actually fewer infected nymphs. So that's awesome. So then the chances of those nymphs going and biting a person and that person contracting disease is much lower where there's more lizards, as opposed to squirrels, which are a reservoir for Borrelia in the system. So speculatively, that's what I think is happening in these desert tortoises, and why that Borrelia prevalence is so low in ticks in the Mojave Desert. And to determine this, so I've started work on this already, again, thanks to the PacVec training grant. I am going to do a borreliocidal factor experiment similar to what Lane did in the 90s. So here's my culture, actually. You can kind of see it swimming around in there. These are Borrelia turricata spirochetes. So instead of using Borrelia burgdorferi, which Lane did, I'm using tick-borne relapsing fever group Borrelia, and in this case, Borrelia turricata. And so I'm culturing that. And then I've also gone and taken blood samples from these desert tortoises. This is actually my dad's desert tortoise. He volunteered to be a part of the study, which that was fun. And I'm taking blood samples from her there. And so then what I'm going to do is I'm going to inoculate these desert tortoise plasma samples with my cultured Borrelia. And I'm going to finish that experiment in November. And I'm really excited to see what happens, because once I inoculate those plasma samples with my Borrelia culture, I watch under dark field microscopy at different time points to see what the spirochetes are doing and if they're dying or not, or proliferating. So finally, as previous literature has mentioned that reptiles may help maintain a low prevalence of disease in these ticks, I'm also working on a model. So I'm interested to model the the system based on the assumption that tortoises may also have that borreliocidal component of their blood. And I should mention that just by focusing on tortoises here, we are missing other important hosts, including rodents, which I did include in this model. Because rodents in other parts of the country are recognized as a reservoir for Borrelia species. So what I've done with these models, so here, let's look at this one first, where the number of hosts is on the y-axis and time is on the x-axis. So I looked at infected tick vectors. So tick vectors that are infected with Borrelia, tick vectors that are susceptible to become infected with Borrelia, susceptible rodent hosts and infected rodent hosts, again, because I'm assuming that these in this system are my reservoir, and then the recovered tick hosts. And in my equations, tortoise populations were kept as a constant number. So according to current literature, it's thought that there's about 1 tortoise per hectare in the desert, which is different from what was years ago before their population started declining, when it was suspected that there was around 100 tortoises, 50 to 100 tortoises per hectare. So they've declined quite a bit, according to some scientists. So using that tortoise population as a constant and considering the chance of a tick coming to bite a tortoise versus coming to bite an infected rodent and contracting that Borrelia infection, I found that as these ticks, these infected ticks here in red come in contact with tortoises, they become recovered. And despite the fact of having a infected rodent reservoir population, those infected ticks will still go down even with 1 to 2 tortoises per hectare in the system as time goes on. I also, just to kind of bring my point home a little bit further, and also to see if this was true in my math, I modeled the number of infected ticks versus a tortoise population. And you can see, as you might expect, as the tortoise population increases, the number of infected ticks go down. So predictions from this model, once I put in my data for my actual data from my borreliocidal experiment, could help improve public health by better understanding zoonotic disease dynamics in the Mojave Desert and encouraging conservation also of the threatened Mojave Desert tortoise. So tick-borne relapsing fever group Borrelia in areas of the West is endemic, but I should mention that most of this data comes from Borrelia hermsii and the vector Ornithodorus hermsii. Very little is known about the prevalence of Borrelia parkeri and Borrelia terracota in the Mojave Desert. And I think to have so many ticks test negative for TBRF group Borrelia, especially considering they were collected from tortoises and have it in an area where someone contracted tick-borne relapsing fever, is kind of interesting. So I aim to get more at this November with my borreliocidal factor experiment. And I'm also going to do some continued rodent sampling in the Mojave Desert because I want to see how these rodents interact with this system as a potential reservoir host. And then finally, I have yet to finish my molecular-based tick identification. I still have a few hundred of those to finish. But I do think that this data will help to manage a disease that is a potential risk for people living in Las Vegas and the Mojave Desert. So with that, thank you for your time, and I'll be happy to take any questions.
All right, really nice, Molly. And this is one of those stories that has so many different angles and a lot of things we don't know about. It's always interesting to hear. Are there questions from the group?
Oh, yes, I have a question. I'm from Humboldt State. So I'm really interested about your borreliocidal experiment. I'm not that familiar with that experiment. But I was wondering whether it's possible Borrelia can die by itself. I understand you mix the culture Borrelia with the blood from lizard. I was wondering whether any control in the experiment to monitor that Borrelia die by themselves in the test tube.
Yeah, that's a great question. So we use mouse plasma because that is a proven reservoir for Borrelia. And in fact, that's what Lane did. Those spirochetes tend to proliferate in the mouse plasma as opposed to just being in the tortoise plasma or the lizard plasma in the case in the 90s.
Okay, thank you.
We have a question from Choki Sherpa from Kern Mosquito Control, who's asking if the two species of Ornithodorus ticks coexist in the same burrows, or did they come from separate burrows?
No, as far as I know, they coexist. In fact, from some of my sequence data, I found them at the same sites as I found them as Ornithodorus parkeri. So yeah, they absolutely coexist.
And Tara Roth from San Mateo is asking, are tortoise burrows used by a lot of different species, or do you expect any of those species might be important for the maintenance of Borrelia?
Yeah, that's a good question. And that's something that's really difficult to answer. That's a question that's difficult to answer because tortoises are keystone species in the fact that they do create habitat for other animals, from owls to other lizards, in fact. But we do find these ticks on tortoises where we don't find them on lizards. But that's where the rodent question comes into play too, right? Because rodents are known as reservoirs in other parts of the country for Borrelia, and which is why I want to do rodent sampling, because I want to see if they are also a reservoir for Borrelia in the system as well. But yeah, there is a lot going on. You could ask questions about the system forever. And I'm just taking a little snapshot at a couple of interesting hosts. Thank you.
Yeah, so cool. Love your study. All right. And then one related question I had was that, of course, tortoises live a really long time, also relative to the rodents you're talking about as well. So I'm curious what immunity looks like for Borrelia. And if tortoises have long-lived immunity, I wonder what that does to their potential for being a host of the Borrelia that would matter.
Yeah, that's a good question. And I'm actually working on doing an immunoassay with some scientists at Baylor College of Medicine in Texas. And because I don't know how their immunology works in the case of a Borrelia resistance. Is it innate? Lane suspected that it was in the lizards, was part of their complement system. In fact, he went as far to say that it's a part of a thermolabile component in their blood, which there's also a thermolabile component in tortoises as well. But if they are creating these antibodies, I want to know. So that's another part of my experiment that I'm trying to determine. So far, we've had kind of hit or miss with the Western blot for the antibodies, but we're still working on developing it.
Okay. And Nikos Gerfield from San Diego is asking, are Borrelia considered endosymbionts of soft ticks?
That's a good question. I don't think that they are, actually. So could you ask that? So are they endosymbionts?
I didn't mean to throw a hardball or anything there. Just, I guess looking at rickettsia and all the various rickettsial species, many of them are considered endosymbionts, that they kind of co-evolved with the ticks and don't really cause any pathology and such. Just did not know if Borrelia had that same kind of relationship with the soft-shelled ticks. So if you don't know, fine, kind of different than tortoises, but just thought of it. Thank you.
No, that is a good question. In fact, a lot of people speculate that they are, and that it's actually the ticks that are the reservoir of this bacterial pathogen and not necessarily rodents. But again, there hasn't been a lot of research, and that's a difficult question to ask and to answer for sure.
Do you know, last thing on that, do you know if they reside in a particular organelle or organ of the ticks?
So they're mostly in the midgut, which is really important for this blood, this borreliocidal factor, because it does kill the bacteria in the midgut. That being said, it can be left on the mouth parts too, but for the most part, the spirochete hangs out in the midgut of the tick.
Awesome, thank you.
Mm-hmm.
Good luck on the study.
Thanks.
All right, so we're at the halfway point for the hour, and Molly, thank you so much. Really nice work. We'll go ahead and move on in the interest of time. And next up, we have Whitney Holeva-Eklund. And Whitney is a candidate for her PhD in the Interdisciplinary Health Program at Northern Arizona University. Whitney's work is in Dr. Crystal Hepp's lab, and she's focused on understanding the relationship between health equity, or perhaps inequities, and the invasive mosquito vector Aedes aegypti in Maricopa County, Arizona. She's hoping that these projects will directly inform mosquito control practices and in Maricopa, as well as potentially other desert regions, by creating a better understanding of how this mosquito is thriving in our arid environments in the Southwest. Whitney defended her proposed dissertation research last fall, and she's planning to complete her dissertation in early 2023, after which she's hoping to pursue a career with CDC. With that, I'll turn it over to you, Whitney, when you're ready.
Thank you for that introduction. So let me pull up my presentation here. All right, so like Chris said, the talk today that I'm going to be giving is focused on Aedes aegypti in Maricopa County and exploring the association between that mosquito vector and some weather and socioeconomic variables. So for anybody not familiar with Aedes aegypti, they're known as the vector of dengue, yellow fever, Zika, and chikungunya viruses. So extremely relevant to public health. So to start out, I just want to kind of go over the broad goals of our project and kind of our long-term goals for the collection of projects that we're working on related to Aedes aegypti. So our long-term goals are really to develop some models and develop better understanding that can kind of inform the range of risk or the potential risk to the public in Maricopa County prior to the occurrence of any outbreaks. And so we'd like to get these models going and really understand how Aedes aegypti is surviving and thriving in this area before any diseases are potentially introduced to the population, to really give us a head start on responding to potential outbreaks. So we've seen in the past a couple of examples of, when West Nile virus was introduced first in New York, it spread across the country really rapidly because the vector that transmits West Nile virus was present in a lot of the country. And so with the established presence of Aedes aegypti in Maricopa County, it's very possible that a disease becomes established in the population very quickly and doesn't give us a lot of time to respond. So we'd like to develop this understanding prior to that occurring. So for this project in particular, we're just trying to explore the association between Aedes aegypti abundance and weather and socioeconomic variables in the desert climate of Maricopa County. And so what we did was compare 3 different statistical models, a baseline model that didn't have any weather or socioeconomic predictors. We had a weather only model, so we just incorporated weather predictors, and then we added socioeconomic predictors to those weather predictors to see if that could improve the model, but really just to kind of be able to characterize those associations and kind of let us kind of figure out what direction we need to go next for a lot of this work. So going into the outline of the presentation today, I'll go through 5 different sections. So the first is some background on Aedes aegypti in Arizona. Then I'll go through kind of what inspired us to start these studies or this collection of studies, the methods that we used for this project, the results, and then some of the conclusions that we found in this project and the next steps that we'd like to pursue. So going into that first section, Aedes aegypti in Arizona is kind of a weird match. So Aedes aegypti is a tropical vector. It generally prefers tropical kind of humid climates. And Arizona is not tropical or humid. It's very dry, it's very hot. In fact, kind of hotter than the vector actually can tolerate a lot of the year. So it's not generally thought to be kind of a natural match between the habitat and the vector. But nonetheless, Aedes aegypti has been identified in Arizona all the way back to the 1930s. And then it's been trapped pretty consistently and routinely in Maricopa County over the last decade, and has started to establish a really strong presence in the region. And that's something that's true for a lot of the Southwest of the United States. So the vectors started to establish a strong presence in those desert regions, but it's also just generally been expanding its territory into more temperate regions throughout the world. So we're seeing this huge range expansion for the vector. And that's true also for Maricopa County. So Maricopa County Vector Control has a really, really great trapping program set up. They employ about 800 routine CO2 traps that they set up throughout Maricopa County on a weekly basis. Most of these traps are located in the Phoenix metro area, since it is a public health service. They're primarily interested in mosquitoes that are around population centers, which is going to be Phoenix metro. So if you're familiar with Maricopa County, it kind of extends down to the left over here. It's kind of cut off in the picture, but that's a lot of rural desert area, not a lot of population. And so the traps are mainly focused in this Phoenix metro area. But really, really great, robust trapping program, provides a ton of data going back years. So we were able to take advantage of that data and use that in this project. So a little bit of background on the actual numbers of Aedes aegypti in Maricopa County going back to 2006. Although this project is focusing on 2014 to 2020, because that's when the vector really seems to have taken off in the county. But if you look back to 2006, you see, a couple of Aedes aegypti that are found in the county each year. And then 2012, 2013, it starts to pick up. In 2014, it really just explodes. And ever since then, the vector has been really established in Maricopa County. You see in 2018, the number is a little bit lower. We ran into some data storage issues where we lost a little bit of the data at the end of the year there. So that number is a little lower than it should be. And then of course, 2021 is not even over yet. So that number is just going to keep going up. It's already the biggest year we've seen on record. And it's just going to keep getting bigger. So no reason to think that Aedes aegypti is going anywhere. It seems like they are in Maricopa County to stay. We see 2020 is also a little bit of a lower year. The reasons for that, not totally clear. That is true for all mosquito vectors in Maricopa County, not just Aedes aegypti, and it may have something to do with the fact that 2020 was a very dry year. But again, that's just a little bit of speculation on our part. And then in 2021, it rebounds right back up. So no reason to think that that is a trend that's going to continue downward. Looks like Aedes aegypti is here to stay. And so this is just another look at that same pattern. The number of traps has changed a bit throughout the years. So going back to 2006, there were only about 285 traps. And then the part of this graph that we are focusing on for this project, it stays about 800 traps per year. But again, you see the same exact pattern regardless of the number of traps that are being used. And again, this 2021 number is just going to keep going up. Excited to see kind of where Aedes aegypti is going from there, seeing as how 2021 is kind of the biggest year on record so far. So now going into a little bit of the background or the inspiration that we pulled from to put the study together. So we are hearing this anecdotal link between Aedes aegypti and higher income areas from the vector control staff. And if you think about it, it should make sense that higher income areas might be habitat for Aedes aegypti. Of course, Maricopa County is generally desert, very dry, but these higher income areas often have golf courses that are watered frequently. They may have water features, more vegetation that provides more shade and can keep the temperature down, keep habitat from it, the water and habitat from evaporating as quickly. And so if you kind of think about it, that should make sense. And so we wanted to explore that anecdotal link and see if we could back that up with the data that we were seeing and that was being collected throughout Maricopa County. We also conducted a literature review and found just 11 articles from mainland USA, and we wanted to focus on just the mainland US because habitats around the world look so different, and so we wanted to really hone in on what that could look like in the US. And we wanted to look at the relationship between Aedes aegypti and socioeconomic factors in these studies, and found some mixed results. A lot of the studies were from Texas, Florida, Louisiana, so a little bit more tropical humid areas than Arizona. A couple of them were from Arizona, and I'll touch on that in the next slide. But we were just curious to see kind of if our findings maybe lined up with those studies from other areas, or if there were some different dynamics happening in the desert. And so the two studies that we found in nearby Tucson, Arizona, one published in 2011 and one published in 2018 by Walker and her group down at the University of Arizona. So they found some positive associations between Aedes aegypti and home ownership and house age, and then a negative association with household income, which is opposite of that anecdotal link we were hearing. So another reason why we wanted to explore this further, because we were hearing one thing from vector control, but then in Tucson, they're finding the opposite outcome. They also found no association between Aedes aegypti and population density, which is interesting given that Aedes aegypti is an anthropophilic vector and has such a close relationship with humans. You would think maybe the more humans were around, the more Aedes aegypti could be in that area. But again, they found no association with population density, another thing that we wanted to explore in our study. And then there was this other study published by Monaghan and his group in 2019. They used temperature and humidity to predict Aedes aegypti abundance in Maricopa County. And they used kind of the whole county aggregated together and a monthly time scale to do this, to do their predictions. And at the end of their paper, they called for the inclusion of some non-climatic factors in future studies. They wanted to take up that call, and our goal in this project was to combine some weather and socioeconomic factors. One, to see if the socioeconomic factors could improve predictions in the model, but two, really just to gain an understanding and characterize those associations so that we could see which directions we needed to go for future research. So that brings me to the third section, getting into the methods of our project and what we actually did here. So like I mentioned before, Maricopa County Environmental Services Vector Control Division has such a great trapping program. We're so thankful we were able to use this trapping data from 2014 to 2020. It just provided us a huge amount of data that would have been really difficult to collect without their help. So we were able to use that data. And so what we did was matched each of the trap locations to the census tract that it was located in. And then we aggregated the number of Aedes aegypti females within each census tract and epidemiological week. Like I mentioned before, the records are close to complete. There were some data storage issues where we lost the end of 2018, a couple months at the end of 2018. And then there's about 2 weeks every year where the vector control staff just needs some time off. And so we're missing about 2 weeks a year, but really, besides that, the records are 2014 to 2020, almost 800 traps a year, still a ton of data that we were able to work with. So the weather data we downloaded from the PRISM Climate Group at Oregon State University. And what they do is take weather stations that are placed around the country, or in this case, we're primarily interested in Maricopa County, and they're able to interpolate the weather values between those stations so that you can have a continuous surface of weather data to work with. And we are really interested in that because Maricopa County, or Phoenix, is such a large land area. We see a lot of kind of differences in the amount of precipitation or temperatures between different areas in the city. And so we wanted to incorporate those differences and kind of get down to a more granular level of meteorological data for this project. So what we downloaded was actually daily meteorological data for the centroid of all the census tracts from 2014 to 2020. We downloaded precipitation, mean temperature, and then mean dew point temperature, which you can think of as just kind of the amount of moisture in the air. And then we summarized weather data for each census tract and epidemiological week. And we were able to match up the Aedes aegypti observations to this weather data, both for the current week of the observation and the 2 preceding weeks, to see if there was some sort of a time lag effect going on there. So for the socioeconomic data, we use census tracts, which are meant to be small, relatively permanent geographic areas with an average population size of about 4,000 people. And they're also meant to be very consistent in these socioeconomic factors. that are present in that area. So pretty consistent in income or house age, population age, those kind of things. So that you're able to use those kind of as a basis of comparison between different, almost neighborhoods really. What we used was the 2019 American Community Survey 5-year data. And we use this 5-year data because it's supposed to be a little bit more stable and a little bit more accurate and reliable than the 1-year estimates. And so we weren't able to, for each year, have a separate census data set, but we were able to use data that's supposed to be pretty reliable and accurate and consistent within each census tract. And so we downloaded this census data for population density, median income, median house age, the percentage of homes occupied by the owner, median home value, the population age, and then the percentage of the population of Hispanic or Latino descent. And all of these variables are either past studies have looked at them and found some associations between Aedes aegypti and these variables, or studies in Maricopa County looking at different vectors have found some evidence of population age influencing the structure of the mosquito population, or CDC guidelines identifying some populations at higher risk of Aedes aegypti-borne diseases. And so all of these variables were kind of grounded in past research or past guidelines, which is why we chose to include them. And so getting into the model building, what we were actually able to do, we used a Poisson generalized linear mixed effects model with a log link. And we incorporated the year, the season, so either mosquito season or not mosquito season, and then number of traps in that census tract as random effects to control for those differences. We standardized the predictors before any of the model building so that we could more easily interpret and compare them later on, since they are all on such different scales, such as like income versus population age. And then what we actually did was an iterative backwards stepwise predictor selection using 500 bootstrap replicates for each iteration to select the predictors. So we started with the weather model first. We added in all of the potential weather predictors, and then we used this backwards stepwise predictor selection with the 500 bootstrap replicates until all of the weather predictors were significant at an alpha 0.05 level. And then once we had that weather model, we considered that our final weather model. We added in all of the possible socioeconomic predictors and selected them in the same way until we reached a final combined socioeconomic weather model. And then we had our final 3 models. So the baseline model, there was no predictor selection. We just had those random effects, the weather model, and then the combined weather socioeconomic model. And for these final models, we're able to do 1,000 bootstrap replicates to estimate the coefficients of the predictors and their confidence intervals, as well as test set root mean square error values to compare the models. And then I should say one advantage of these predictions being at the census tract level is that not only were we able to look at it at the census tract level, but we were also able to aggregate those census tract predictions to a whole county level to get a more zoomed out view of what was going on week by week in the county. So we were able to look at it in both those ways. So the census tract week and then county week as well. And that's really helpful because a lot of really the only past studies in Maricopa County have been focused at that county level. And so we wanted to incorporate that as well so that we could more easily compare our model to theirs. All right, so going into the 4th section, the results of our study. First, I'll just show you some of the graphs for the model fits. So this is looking at the 3 different models. So the baseline model, the weather model, and the combined weather and socioeconomic model. And on the top row, you see in black is the actual observed values, and in red is the predicted values. And on the bottom is just the residuals. So just the observed minus the predicted for each of those models. And so for the baseline model, you can see because of that random effect for season and year, you see the this kind of pattern where it's an off season, an on season, and then the years are slightly adjusted based on whether that was kind of a low year, such as 2020, or a higher year, such as 2014. But you're not seeing those granular kind of week to week shifts in mosquito activity with this model, obviously. And then for the weather model, we see kind of a closer fit in some areas where we're kind of tracking along with the kind of week to week changes in mosquito abundance. But a lot of places we're still missing quite a bit. So this like beginning of 2014 here, we're missing quite a bit. And then just some of these spikes we're not quite capturing. And that's true also for the weather socioeconomic model, which looks pretty much the same as the weather model. So those socioeconomic predictors don't add a lot of predictive power to our model. And we see we're missing in the same places, missing some of these higher points. And that's, so the similarities between the weather and the weather socioeconomic model are reflected also in the root mean square error comparison. And so each of these values is the mean of 1,000 root mean square errors that we calculated from the bootstrap replicates. And so when we're looking at the tracked error, you see there's not really much difference at all between the three models. When you're looking at the county level error, so after we've aggregated the tracked predictions, you see that there's a little bit of separation here between the baseline model and then the weather and combined models. The weather and combined models are almost identical. And so since we're interested in interpretation, we'll move forward with the combined model, but with just this in the back of our minds that there's not a huge improvement in the model because of these predictors. And so looking at the combined model coefficients and their relationships with Aedes aegypti, and all of these values are based on the mean of 1,000 bootstrap replicates again. So you see, we can say the current week precipitation, if that increases by 9 millimeters, we see about a 5% decrease in Aedes aegypti female abundance at the tract level. Whereas with the previous week precipitation and 2 weeks prior, if that increases, we see an increase in Aedes aegypti abundance. And so this model can't really tell us why these associations are present or are occurring, but we can kind of start to speculate a little bit and figure out what directions we want to go in for future research. And so one of the ideas we have here is just if it's raining, Aedes aegypti may not be able to travel or fly as well, and therefore aren't being trapped as easily, whereas rain in previous weeks could increase habitat for the vector and increase their populations. Some of the more interesting socioeconomic, so down here in orange, the socioeconomic predictors. So population density, we see as that goes up, Aedes aegypti abundance also goes up, which we expect. Median income, as that increases, Aedes aegypti abundance increases as well. So that is confirming that link that we are hearing from vector control staff. So I won't go through all of them, but some interesting relationships there and some things that we'd like to explore further in the future. And then we could look at the tract level. So this is just zoomed into the last 3 years because the tracts are so small, it's a little difficult to see. And this is just highlighting, so the mean weekly residual for each census tract for each of these 3 years. And you can see that there's some census tracts that have much higher residual values. If you look kind of the southeast, there's commonly some tracts with higher census or higher error or residual, I'm sorry. And so we'd like to explore why those tracts have that higher residual and what's going on there a little bit further. So now getting into some of the conclusions, next steps. So our aim was to really just to better understand this relationship, and all the differences between models were very small. This has allowed us to begin exploring the relationships and point us in the right directions. And of course, the study had some strengths and limitations like all do. So some of our strengths were just the large amount of trapping data that we were able to harness thanks to the Maricopa County vector control. And then the smaller temporal and spatial scales that we use. So looking at the week and census tract level versus county and month time scales. And then of course, limitations. So the trapping locations were not randomly placed. They were placed in areas meant to trap more mosquitoes so that they can be tested for disease and contribute to greater public health. And then the socioeconomic and weather data were not directly recorded, which you could kind of look at as both a strength and a limitation really, because it allows us to continue using this model and updating it as years go by without having to do some really intensive on the ground work. But of course, we're not getting the socioeconomic data for the houses that are directly surrounding a trap. One of the other issues that we dealt with was some really, really large observations of Aedes aegypti. And these are an example of a few of those with the surrounding area. So most of our observations were just a few mosquitoes, less than 10 mosquitoes at a time. And then occasionally we get just huge amounts of Aedes aegypti. So it's really unclear whether these are some source locations or whether the mosquitoes are attracted to these locations. So we'd really like to start exploring that a little bit further, figure out why we have these really large observations. So I think I've kind of touched on it, but some of our next steps are looking at land cover and that's relationship to mosquito abundance, so greenness or different land uses. We'd like to look at the movement of Aedes aegypti around the county, how far their range is, how they're moving between different areas, and then look at whether there's some source locations that are feeding the population throughout the county. And with that, I just like to thank and acknowledge all the people that were involved in this work, my committee chair, Dr. Crystal Hepp, and committee members who helped with the project. Of course, Vector Control Division from Maricopa County, because this project definitely would not be possible without all of their very hard work. The PacVec training grant that we got that supported this work and then led into the NIH F31 fellowship that I received earlier this year, and that is going to allow us to continue looking into some of these future directions. So thank you for your time, and I think with that we have a few minutes for questions.
All right, thank you, Whitney. Another great talk, and certainly an interesting topic to many on this call. I see Nathan has a thumbs up, not a hand up. Okay, so if anyone has questions, feel free to unmute yourself and ask them. All right, I'll start with one. I was just curious for the predictors you looked at, especially maybe temperature and income. You saw a positive trend for those two variables in particular, and I was wondering if you saw any evidence that they decline at certain levels. Did you look at whether the relationship's not necessarily linear on the log scale or might reduce at high temperatures or high incomes?
Yeah, that's a great question. So for this project, we just started out kind of looking at those linear relationships, but I think that Phoenix is so hot that that's a direction that we need to go in next, is that potentially there are some days in the middle of summer where it's just too hot for the mosquito. And so I think that's one of the directions we'd like to kind of take it, is to see if we do see a non-linear relationship between the vector and temperature, but we haven't explored that yet.
Okay, in our study in LA, of course, you get outliers on the income scale too, where I think aegypti is not necessarily a problem of the rich. It's somewhere in the middle. So it'd be interesting to see that relationship as well.
Definitely, agreed.
All right, Nikos.
Did you look at like the factors of control efforts, if that could have affected the data, and between different socioeconomic areas or whatnot, and if that's a confounder? The intervention of Maricopa vector control, which I think is fairly aggressive, but I'm not sure.
Yeah, I think that's a good point. We didn't incorporate it into this study. And I think it's a little difficult with Aedes aegypti because they are such like backyard vectors, where vector control doesn't really have access to some of those areas where they may be breeding. So like flower pots in people's backyards, that kind of thing. So I think a lot of the spraying that they do is focused on kind of drainage ditches or like parks, that kind of thing, where Culex mosquitoes breed more frequently. And so I think it's something very important we should look into incorporating in the future. But it's, I think, a little complicated when you're talking about Aedes aegypti.
You might look at if their control has certain triggers for intervention, i.e. they get a trap that has 10 or 20 Aedes, or more there. Does that mean, okay, we're sending out control staff and controlling that, versus, okay, well, we see a low level, in these traps, we can't respond to 800 traps like that. So, do they let the low trap counts go, or maybe get to them at some later point in time? I just see that as a possible confounder to what's a great data set, and just looking a little further to enhancing some of the associations you might be thinking you're seeing there.
Yes, yeah, definitely agree. I think, I mean, there's just so many projects I think that we'd like to do next, and so many ideas. And I think that's another really great one that we'll be working on. So.
Hey, this is John. I'll answer a couple of those questions. We do have a threshold of 50, and we'll go in with handheld foggers to treat. But most of those big catches that you're seeing are coming out of storm drains. And that's where the Aedes aegypti hang out during the daytime because it's so hot. It's hot even around, highly vegetative areas. So they go down underground, and that's where they hang out. And that's where you're seeing those big catches of, 1,200 plus in one day. And we'll go in with a handheld fogger and hit those as needed.
All right, Whitney, we have one more question from Choki Sherpa, who's asking, given that the difference between the weather model and the combined weather and socioeconomic model was minute, is it reasonable to conclude that socioeconomic variations don't matter much?
I think that's, yeah. So I think that's a good point. There isn't a huge difference. They don't make the model worse, which is why we moved forward with saying, we can still try to start to interpret. But I think what we're trying to do, or what we ended up kind of concluding with this study, is that these aren't really final kind of set in stone conclusions, but maybe some directions that we'd like to explore further in some more like detailed on the ground research. But we needed, some place to start. And so we started kind of more broadly. And now that we have those directions that we'd like to explore in a little bit more detail, that's where we're going to go next. So I think that's a great point, and definitely something to kind of interpret everything with a grain of salt. But I think for our purposes, kind of just getting the directions that we need to go next, this was still kind of a helpful exercise.
All right, well, thank you so much. And thanks to the group for really nice discussion. So keep those questions coming in our future seminars. We've got another one coming up in 2 weeks, I believe. I should have the date in front of me, but I don't. You'll get email reminders as usual. And I really want to thank Whitney and Molly today for 2 really great talks, and also some very productive training grants from the PacVec perspective. So look forward to working with you and watching your careers as you go. And with that, we'll say goodbye for today and see you all next time.